Hello everyone, today's topic is ELISA or ELISA. So, let's see what is ELISA. It is an enzyme linked immunosorbent assay. Here the term assay is related to something analytical, right? In case of ELISA, we can process an analytical assay or analytical process by which we can identify or we can determine the concentrations and the types of the antigens or antibody, right? So, let us see its discovery. First discovered by Engels and Parmelan in 1971. Firstly, it is used for detections of various fungi, viruses, bacteria or other microbes which are responsible for the disease. Okay. Basically, we can say for disease identifications or disease causing organism identifications, we can use the ELISA or ELISA process. Let's see. As I said before, it is a qualitative and quantitative analysis by which we can identify or we can determine the concentrations of a specific antigen or antibody. Also, we can determine the types of antigen and antibody. Which types of antibody is responsible for specific disease, we can identify through this ELISA process, and also we can determine the disease specificity. Disease is acute or chronic, the concentrations of the antigens, specific antigen presence in the samples or in the body. Okay. Let us see what are the types of ELISA or ELISA. There are 4 types of ELISA, that is direct ELISA, indirect ELISA, sandwich ELISA and competitive ELISA. Let us see the first process, that is direct ELISA. So, this direct ELISA also called simple ELISA as because the process is very simple, mainly used for the detection of antigens which may be proteins or may be peptides or may be hormone, right. Direct interaction between the enzyme linked antibody and antigen process involves. Here in case of direct ELISA, you can see there is a term direct. So, it is direct interactions between the enzyme linked antibody and the specific antigen. So, when we start some ELISA process, any types of ELISA process, we need to know about the previous process, that is collection of blood or collect the blood sample. Then we isolate the serums. Serums means where the antibody or antigens are present. So, after isolating the serums, we have to reserve it. Reserves in phosphate buffer saline. Phosphate buffer saline is a proteins, right. PBS, its short forms is PBS, phosphate buffer saline, closely mimic the pH, osmosity and ion concentrations of the human body. That means this phosphate buffer maintained the inner conditions of our body. Okay, that's why we can assure that sample serum is absolutely perfect, or we can say that our sample is in perfect condition, right. So, what happened in this direct ELISA or steps involved in the ELISA, direct ELISA? Let us see what are the steps. First of all, we need to add the sample which is contains with the specific antigens, right, which I have to determine, right. Next, the surface blocking. Then the surface blockings with the help of bovine serum albumin. These are proteins, non-reactive proteins which cannot react with the antigens and other organisms. That's why the bovine serum is used for the surface blocking. Why surface blocking is needed? As because there is some empty places after entering the antigens into the microtiter place. We need to block this open surface. That's why we use the surface blocking agent, that is bovine serum albumin, a non-reactive protein. Next steps, that is introduced enzyme linked antibody into it, which is previously designed for these specific types of antigens. Then after some times we need to wash out the floating antibody, those antibody which are not attached with the antigen, right? We need to wash them. That's why washing process is needed. Next, the 5 steps, specific substrate introduced, which can be binds with the specific enzymes which are already present in the where the enzyme link antibody. Here you can see in these pictures there is a triangle like shapes are the antigen, specific types of antigens. After that there is a enzyme link antibody. Red shaped symbols are indicated as enzyme, and this is an enzyme link antibody. After this we add something, that is the substrate. It's also called chromogenic substance. Those chromogenic substance attached with the enzyme link antibody, and they form a complex with the specific types of enzymes. The chromogenic substance may be the horse radish peroxidase, beta galactase, alkaline phosphatase, etc. Then these substrate and enzyme reactions causes some colorations. Those substrate enzyme combinations give a color, and the intensity checked by the spectrophotometers or spectrophotometer like instrument, right? So let's see in these pictures there is the antigens, and here the enzyme link antibody is present, that is HRP, that means horse radish peroxidase linked enzymes is present in this case, and after that the chromatin substance is added, that is TMB substrate is added, and it forms a blue color. After this color formations, we can check with the spectrophotometers and identify the specific types of presence is present or not. If there is a color formations, so that indicate the presence of specific antigens in the sample. If there is no color formation, that means the test is negative, that means sample is free from this antigen, right? That's all for the direct ELISA. Let's see the indirect ELISA. It is specially designed for the antibody detections. First of all, we need to ready-made microtiter place which is already fixed with the captured antigen. That means for specific types of antigen there is a presence of some specific types of microtiter plate. That's why we call this ready-made microtiter plate. Microtiter plate are disease specifics. As I said, microtiter plates are disease specific as because they have already fixed with a captured antigen. It may be specific for malaria, it may be specific for dengue, etc., etc. So next step, that is the sample antibody, that is called primary antibody, binds with the antigens. Then we add the sample which is also contains with antibody for specific antigens, right? When some virus is attacked in our body, then those virus are replicate themselves, right, or copying themselves. During this copying times, our body already creates some antibody. Those antibody are present in the sample. So sample antibody, that is called primary antibody, binds with the antigen, right? After some times, second antibody, that is enzyme link antibody, is introduced into it, right? Those specific types enzyme link antibody also specified for this antigens which is coated into the microtiter plates. So next steps, that is the 4 number steps, is substrate attached with the enzyme and give color product. Obviously those enzymes which is introduced in this process, that is the enzyme link antibody, that means a antibody which is linked up with the specific enzymes. So for the specific enzymes we have the substrate, specific substrate, that is chromatin substance, chromogenic substance. We need to attach or need to add those for the special coloration. Point to be noted, you have to wash every times when you add a specific type of enzyme link antibody and also specific type of substrate. So, let us see next steps, that is wash free substrate. Those substrates which are not binds with the enzyme link antibody, we have to wash them. That is why we need some washing process. Those after some times, those substrate and the enzyme link antibody from some product which is colored, right. So, here colored formation is occurs. Then we need to identify the intensity by the spectrophotometers or any other mechanisms is followed for the intensity check, right. That is all the indirect types of ELISA. Next process is what? That is sandwich ELISA. So, what is sandwich ELISA? Here the sandwich terms something in between, right. So, in case of sandwich ELISA, we can determine or detect the antigen. Especially for antigen detections, the sandwich ELISA is preferred. Antigen is sandwiched between two antibodies. Here antigens is determined by this process. Those specific antigens is sandwiched between two antibodies. That is why this process is called sandwich ELISA. The sandwich ELISA quantify the antigens between two layers of the antibody, that means captured and detections antibody. Two types of antibodies are here, one is captured, another is detection antibody. So, two different antibodies, either monoclonal or polyclonals, that means antibody have monoclonal activity or polyclonal activity. Any antibody can be used that are reactives with the different epitopes on the antigens which with a concentration that need to be determined. That means two different types of antibodies are used, either may be monoclonal or polyclonal, that are reactives with the different epitopes, right. They have a property to react with different epitopes on the antigens with the concentrations that needs to be determined. So, let us see the steps involved for sandwich ELISA. There is a coated antibody are fixed with the microtiter plate. As I said before, those microtiter plate are disease specifics. That means those microtiter plate have coated antigen in it, right. Coated antigens, it may be coated antigens or it may be coated antibody which are specific for a specific disease or special disease, right. That is why we called coated antibodies are fixed in the microtiter plate. In this case, sandwich ELISA, there is no antigen is coated into the microtiter plate. Antibody, specific type of antibody is coated into the microtiter plate. Next, the sample with antigen introduced into it. Samples means the serum in which the special types of antigen is present, right. Then next steps is unbound antigen washout. After entering into this microtiter plate, the antigens binds with the specific types of antigens which is coated into the microtiter plate. Sorry, antibody which is coated into the microtiter plate. Specific antigens, they binds with the specific enzymes or antibody, right. So, there is some unbound antigen also which cannot binds with the microtiter plate coated antibody, right. We need to wash them. That is why we call that unbound antigen washout. Next steps, that is after washing out, a second enzyme conjugated antibody specific for a different epitope of the antigen is added and allow it to incubate. Next steps, what happened? A enzyme link antibody, as you can see in these pictures, the green color antibody which is linked with the yellow color enzymes is added, and those enzyme link antibody is attached with those antigens, right. They only bind with the different epitopes of the antigens, right, not the same epitope. After a specific times or incubation, after incubation times, then specific substrate is added. Here you can see in the green color oval shaped substance is called the specific substrate, or the chromogenic substance is added into it. Next step, the substrate and enzymes produced a color product. As because the substrate is chromogenic in nature, that's why they produce a color product. And the next process are same as like direct and indirect ELISA, that is the wash the free substrate. We need to wash out for the unbound substrates which are present in the microtiter plate. Then the colored formed product is identified or intensity checked by spectrophotometer. So by this process we can identify the specific antigens which is responsible for a specific disease. So let us see next process, that is the competitive types of ELISA. It is quite different from other types of ELISA, that's why this is called as competitive ELISA. It also known as inhibition ELISA. That means in this process there is a competition in between two things, right? Let us see, the sample antigen competes with the antigens that is introduced as a reference for binding the certain amount of labeled or specific antibody. That means there is a competition between two antigens for a specific antibody. Oppositely, it competes with the sample antibody and refers antibody or enzyme link antibody for binding with the coated antigens. That means here is a competition between two antibody for one antigen, right? One types of antigens. It is used for antigen and antibody both detection. In case of both detection, we can use the competitive ELISA or competitive ELISA. So let us see what are the steps involved in this competitive ELISA. Let us see the steps. First is antigens, that is reference antigens are coated with the microtiter plate. In these steps we are following thus antibody competitions, or you can say competition between two antibodies for a specific antigen, right? Let us see what happened. Antigens are coated with microtiter plate, that means antigens which are reference, which act as a reference, right? We all know the samples and antibody combines with each others and they form a complex, right? So next steps we can see sample with within it specific antibodies are present with it a specific amount of E-link antibody. That means samples which is already contains with a specific type of antibody, within it we can add the enzyme link antibody or fixed amount of enzyme link antibody. Next steps, add it into the microtiter well. We need to add those sample which is also included with enzyme link antibody and the antibodies which are present in the sample need to be added in the microtiter plate or microtiter well. Antibody compete with each other to bind with antigens. Obviously, the microtiter plate have the specific types of antigens and we add something, two different kinds of antibody, right? One is enzyme link antibody which is in fixed concentrations, and the specific types of antibodies which is also present in the sample. So there is a presence of two types of antibody, right? The antibody compete with each other to bind the antigens. Obviously, two types of antibody trying to compete with each other for binding with the antigens. Then next process is wash out free antibodies. Those antibodies which are free, then we need to wash out them, right? Next steps, that is chromogenic substrate added into it. Chromogenic substance means it may be horseradish peroxygenates or beta galactase, or it may be the alkaline phosphorus. Any types of chromogenic substrates we can add into it. So for this specific chromogenic substance we have to some specific types of enzymes which is already present into the enzyme link antibody. So next steps is washing out or washing again for free substrate out. Those substances which are free after binding with the specific types of antigens, we need to wash them. That's why the washing process is needed here. So next steps is number 7. Intensity can be determined by spectrophotometer. After binding with the enzyme, those substrates gives a color product. Those absorbency is checked by these spectrophotometers or as like spectrophotometer machines, right. Next is if absorbance less, that means the sample antibody is more. If absorbency is more, that means the sample antibody is less. Obviously, those antigen linked antibody which is linked with an enzymes, these special types of antibody is binds with the substrate and they get the product. Others, the sample antibody cannot binds with the substrate, so it cannot produce the color substance. That's why we can say if absorbency is less, that means the sample antibody is more. Less colors mean less sample antibody. More colors means more fixed amount of enzyme link antibody. So we need to determine the sample antibodies. That's why we can check the absorbency is less, means the absorbency sample of antibody is more, and the absorbency is more. In that case, the sample of antibody is less. Antibody with greater concentrations mean the competitions. Those antibody which have the concentration greater concentrations, those antibody win this competition, right. Let's see a picture about this. Here you can see in this picture, green color substance is chromogenic substrates and the red color is antigen, this yellow color is sample antibody, and this is the enzyme link antibody. When a microtiter plate is used, those specific types of antigen is already attached or you can say coated into the microtiter well. After that, we add sample and the enzyme link antibody into the microtiter plate. After adding this, those specific enzyme link antibody attached with the antigens and or you can say both the sample antibody and enzyme link antibody compete with each other and they binds with the antigens. And then we add the chromogenic substrates which is binds with the specific types of enzyme link antibody. As you can see in these pictures, the specific substrate are attached into the specific types of enzyme link antibody and others is as usual same. So here you can see in these pictures, here is a enzyme link antibody is win this competition. That means those samples which we are used are less in concentration. That's why they produce less absorbency or they produce less color after this competition or after this competitive ELISA. So that's all for this ELISA. It is included in competitive ELISA, sandwich ELISA, direct ELISA, indirect ELISA. Hope you all are satisfied for this topic. Thank you for watching. If you like this video, please do like, share and subscribe.